Alright, so now we're going to talk about DNA replication, which is actually S phase in the cell cycle, which we talked about previously. So, in cell cycle, we have the first growth phase, and in the growth phase, the cell is getting bigger, it's taking on more organelles or producing more organelles, and critically, it's producing ATP energy. Now, that ATP energy is going to drive the process of splitting all of these chromosomes, literally unzipping them down the middle and adding a whole new piece of zipper all the way along both sides. So, the process of DNA replication in physiology is not absolutely and utterly germane to what we're looking at in physiology, but having an appreciation of how it works is helpful to us. So we'll go through it in very basic terms, and then after that we can therefore look at protein synthesis based off what we've learned about DNA replication. Okay, so on the board here I've got DNA replication, S phase in the cell cycle to remind us where we are in the cell cycle. And if you can think back to week 3 when we talked about macromolecules, the last of the 4 was the nucleotides. And a nucleotide is made up of 3 parts. The phosphate molecule, which is a phosphate surrounded or bonded to oxygens with lots of negative charge. This is bonded onto a ribose sugar, a 5-carbon sugar or pentose sugar. You'll see it written in all these ways in textbooks. And that's got the pentagon to show that the 5 carbons. Phosphate. Now, the phosphate sugar makes the backbone, or one rung of the ladder. I mean, basically DNA looks like a ladder, 2 backbones with the PS, PS, PS all the way down on both sides, etc. So that's our phosphate and our sugar, and then sticking out as half of the rung is the base. Now the base is a nitrogenous base, so it contains nitrogen, carbon, hydrogen, oxygen, and there are 4 types that you find in DNA. Now earlier I said the sugar was, I said it was pentose sugar, 5-carbon sugar, and I said ribose. Now I misspoke, because this is DNA, so it's deoxyribose sugar. And that's one of the differences between DNA and RNA, is that there's one less oxygen on the sugar in DNA than there is in RNA. So it's called deoxy, meaning one less sugar, ribose relating to the sugar itself. So this is a deoxyribose sugar base. We've got adenine, thymine, cytosine, and guanine. For some reason I decided not to put the E in on cytosine. So these are the 4 possible bases. Now when we talk about protein synthesis, we're going to talk about languages. And the English language uses 26 letters to form words, and words form sentences, and hopefully some of those sentences even make sense. The language of our genes is spoken in just the 4 letters, just the A, T, C, and G. So it obviously has a certain limit to how many words it can make, but it's enough to produce literally up to 90,000 different proteins. So it's certainly quite versatile. So we have our nucleotide structure, and we see how the nucleotides orientate with a phosphate sugar, phosphate sugar, phosphate sugar backbone on both sides. It runs in opposite ways. You can see the pentose pointy bits on the top here and on the bottom on the side. So you take and kind of reverse them, and it works that way. We could call it 5' to 3' end if you're getting into the genetic side of things. But we're not, so we'll just ignore I said that. Alright, so we've got the bases pointing out into the middle, and they join together. Now what holds the rungs of the ladder together are our friends, the hydrogen bonds, if you remember, formed between a weak attraction between a partially charged hydrogen on water and on another water molecule. Well, there are hydrogen bonds formed on the adenine and between the adenine and the thymine, and there are 2 in this grouping, and there are 3 between cytosine and guanine. Now I've also gone ahead and drawn them slightly differently. So you'll notice that the A has like 2 bricks and the G has 2 bricks, but C and T just have 1 brick. This is representing the fact that they're cyclical structures, and adenine and guanine have 2 cyclical structures pushed together. So there are 2 bricks. So they are 2 cyclical structures wide, whereas thymine's just 1 cyclical structure wide. Cytosine just 1, and then guanine again has 2. Now they always join up A to T, C to G. You could turn that around and say they join up T to A, G to C. Doesn't matter. A and T always group together, C and G always group together. There's a couple of reasons for this. One, they match up for hydrogen bonding. The 2 hydrogen bonds formed there fit. The 3 hydrogen bonds between C and G fit. If you try and get C and T together, that's 3 hydrogen bonds in a different place, so they don't fit well. The same is true for G and T and A and G, etc., etc. The second reason is that the ladder, a structurally sound ladder, is straight down the sides and the rungs in the middle. Now, if you grouped A and G together, you'd have 4 cyclical structures. So instead of it being straight like this, it would go down straight and then punch out there, where the A and the G we're trying to put together. That's 4 versus 3 there, and then 4 there. And then if it was immediately followed by a C and a T, it would come in really narrowly, C and T, because that's only a 2. And if it came out to a 3, that would not make a structurally sound molecule. And keep in mind that this ladder structure is then twisted on its ends to form a helical shape. That would not twist correctly either. So A to T, C to G is how it works between the nucleotides. So that's how DNA is formed. Now each one of these bases forms part of the code that becomes, the blueprint for a protein. Now, one of the nice things about DNA is that it's got these 2 strands and they're joined in the middle and then twisted on themselves. That's then folded up into a chromosome and wrapped around histone proteins. So it's supercoiled. That really protects the language. It's like taking a book full of important information and putting it in a lockbox, and then taking that lockbox and putting that in a safe, and then from the safe you put it in a building and you put guards around the building. That keeps the letters here extremely safe from being changed by radiation or toxins, chemicals, etc. However, there comes a point, such as S in the cell cycle, where all of this has to be photocopied. All the pages of the book have to come out and be photocopied to create 2 new copies where there was 1. So how do we do this? Well, there are 2 enzymes involved, and you met enzymes a couple of weeks ago in the course, so you now know that enzymes are proteins that catalyze reactions. Well, helicase, A's telling you, and A's there, tells you it's an enzyme, and what comes at the start tells you what it works on. So this is a twisted helix. We've unwound it because it's complicated to try and draw something in a helix, and my art is not that good. So this is the helix unwound. Helicase breaks the hydrogen bonds holding the rungs together, so it unzips from the middle. So that action of helicase exposes the bases. Just realised I left it blank, and of course I put the base in the wrong place. Alright, so the helicase has opened it up and unwound it, so these run and open up all the way along. Once that's done, free nucleotides, which is just the phosphorus, deoxyribose sugar and the base, can come in and can line up in complement to whatever is exposed. So if you've got an adenine base exposed, a T or thymine free nucleotide will come in and line up, and hydrogen bonds, the 2 of them, will form. The same is true on this side with the T being exposed. goodness sake, David, my brain seems to have gone to sleep. And adenine, need to make that bigger, will come in and it will also form a hydrogen bond and bond on. This will continue down the whole line. These would be opened up and they would also, until the whole thing is you've got two new strands. So there's a couple of things to learn about this. One is that one side of the strand makes one new molecule and the other side makes another new molecule. So the original molecule is actually 50% of the new molecules is made up from the original and 50% is made up from free nucleotides. What organizes this happening? Well, as helicase has broken it open, polymerase, polymer to make something big from small, has joined them together, joined them all the way along, and created the two new strands. So polymerase has made these hydrogen bonds, got them lined up in the correct sequence, and now we have two where there was one. And that's what happens in S phase. There's lots of other things to talk about in relationship to DNA replication, but for us as physiologists, that's enough. So thanks for listening.